I'm Meredith. I'm a dietitian. Today I'm going to talk about nitric oxide and how I think that it's a Goldilocks nutrient. And I think that excessive amounts of nitric oxide are one of the contributing factors to all the nasty cascade from hypervitaminosis A. And this video isn't just for somebody who has problems with hypervitaminosis A, but anyone who's experiencing chronic fatigue or MS or autism. So I think that nitric oxide plays a role in all of these particular conditions, or potentially even chronic fungal infections. All of these things can actually be an imbalance in nitric oxide in the body. And so in this video, I want to talk about how retinol binding protein 4, the carrier for vitamin A in the blood, contributes to too much nitric oxide. And then also how metabolic acidosis could be worsening our nitric oxide levels to a point where it's causing damage. And then chronic bacterial and fungal infections can also be causing this issue as well. And why I think this is important is I think that nitric oxide is the thing that is inducing a functional B6 deficiency, and that's leading to the inability for people to recover from hypervitaminosis A. So before we talk about retinol binding protein 4 and vitamin A, we have to kind of understand that not all nitric oxide is bad. We need some. We need some nitric oxide in the body, and it's really a chemical signal. With enzymes, it can help to turn off enzymes when we don't need them, but also nitric oxide can help to stop viral replication, and it can actually kill bacteria. So we need it. We do need it. It's just that at times we can have so much that it's wreaking havoc on our own bodies instead of just on the viruses and the bacteria that we're encountering. So whenever we have a bad bacteria, whether it's a bacterial infection in the ear or in the gut, there's a bad bacteria, it produces lipopolysaccharide. And some of the bacteria in our gut produce more of this than others. And so LPS connects with, on a macrophage, a white blood cell. It actually it comes in here at toll-like receptor 4, and that causes a signaling cascade that includes nuclear factor kappa B, which goes to the nucleus, and between that and P38 and the cascade from that, you actually induce nitric oxide synthase 2, also known as INOS. Okay, and so another factor that happens is when there's inflammation, there's a production of interferon gamma. And interferon gamma enters a different pathway, the JAK-STAT pathway, but it has basically the same cascade that ends up increasing the production of nitric oxide synthase 2, or INOS. So then the enzyme INOS takes oxygen and arginine and uses those two things to make nitric oxide and citrulline. And so normally nitric oxide can play a good role in the body to help fight bacteria and viruses. The problem is in hypervitaminosis A is that RBP4 becomes one more source of inducing nitric oxide. So here's an article that shows all the things that retinol binding protein 4 can do to wreak havoc on the human body. So we're going to be dealing with this one over here, inflammation, but you can see that there's RBP4 has been blamed, rightly so, for insulin resistance in adipose tissue. It's also blamed for beta cell dysfunction. It's blamed for the inhibition of insulin signaling in the muscle. It can also prolong gluconeogenesis. And then it can cause endothelial and mitochondrial dysfunction and oxidative stress. So I actually have a separate video where I think part of the mitochondrial dysfunction is actually related to this thing called A2E, which is a lipofuscin that actually pushes cytochrome C oxidase out of the mitochondria. It just prevents the respiratory chain from working. So RBP4, it's like my nemesis. It's my nemesis because if you don't know, my 12-year-old daughter has hypervitaminosis A and is quite sick from it. And most of my long-term clients have this issue too. So here is RBP4 in relation to inflammation. And so this cascade happens in white blood cells. So RBP4 uses the toll-like receptor 2, and then this toll-like receptor 4 also, similar to LPS, to lipopolysaccharide. And so it enters this way, it causes the cascade to increase P38 and nuclear factor kappa B. And then that in turn comes back through here and increases the production of these inflammatory factors. Okay, and so it will increase TNF alpha, it'll increase interferon gamma. And remember, interferon gamma also works with the JAK-STAT pathway, and so it also increases iNOS production. And then we also know that RBP4 can, induce iNOS production too. So let me show you that. So I did a little modification here based on some literature that I read, and that those references will be in the videos, the video description. But basically, you will have this increase from RBP4 specifically, you'll have an increase in TNF alpha, interferon gamma, and also iNOS directly, as well as, so this TNF alpha can come back out and have a feedback mechanism where it continues to induce nitric oxide synthase. And so what we end up with is a very high nitric oxide state. So what would that look like in someone that, a lot of people are like, well, what does that do? Well, symptomatically, that person's going to have problems with their blood pressure. They're going to have orthostatic hypotension. So going from sitting to standing is going to make them feel woozy. So they might also have more asthma. It might be harder for them to recover from respiratory infections, stuff like that. So I'm trying to think of anything else. And then the big one that I've noticed among my clients and my family members is massive problems with anything B6 related. So it could be elevated liver enzymes, because liver enzymes require B6. And if you're not getting enough B6, your body will just make more of these enzymes to try to compensate. It could be problems with histamine tolerance, like you have really bad allergies, totally stuffed up all the time, hot flashes from too much histamine, can't sleep all night. You could have neurotransmitter dysfunction. So it's a massive cascade. And I think it all goes back to this unregulated amount of nitric oxide. So besides RBP4, most of the people that I've seen on their lab work, they end up having metabolic acidosis. And I think that's because there's a dysfunction in their ability to run energy metabolism. And so overall, they have less CO2 production, but also they're struggling with transsulfuration, which is B6 dependent. And so they make less CO2 in that pathway too. So overall, these people have less CO2, and if you have less CO2, that means you have less bicarbonate, because they kind of go in between each other. And if you have less bicarbonate in your blood, then you're in acidosis. So there's this journal that talks about metabolic acidosis. It's a cardiology journal article. And they say that anything less than 25 is not a good idea for your bicarbonate level or your CO2 level. So in the US, we check bicarbonate by looking at CO2 in the blood. In Europe, they use bicarb or HCO3 levels in the blood. So, or that's how they label it on the labs anyway. So if it's less than 25, you're considered to be kind of in a chronic state of not good pH. If it's less than 22, you're in metabolic acidosis, and laboratory parameters are insane. So like for my own daughter, her CO2 level on her lab is 20, but it's not flagged as abnormal until it's 19. But she's walking around in an acidotic state. So what does that do? Well, nitric oxide can be produced non-enzymatically at a low pH. So we don't even need iNOS. When our pH is low, our body will take nitrate sources and make nitric oxide. So if CO2 is less than 22 on a comprehensive metabolic panel, you definitely need to be careful with what you do with your diet. One thing you don't want to do is you don't want to consume high dose vitamin C, because that actually further increases this production of nitric oxide from nitrate. So that's the thing you have to watch in your diet if you're in metabolic acidosis. If you're in metabolic acidosis, you don't want to eat a lot of high nitrate foods. And even in the absence of vitamin C, this still occurs. In the absence of vitamin C, you still end up at a low pH making nitric oxide just spontaneously. So a low nitrate diet would be beneficial in someone with vitamin A toxicity or hypervitaminosis A, or even chronic bacterial infections, and chronically low CO2 on your CMP. You want to try to eliminate high nitrate foods. And I have a solutions page about that. But obviously, like some of those would be like beets. Celery is very high in nitrates. Lunch meat, sausages, anything that has nitrates in it, even if it has celery powder. Celery powder is not benign. Just because it's natural nitrates doesn't mean it's nitrate free. It actually still has nitrates. So eating more fresh animal products would be better. And definitely avoid juicing things, juicing celery, juicing beets. Those things are going to raise your nitric oxide level if you're in this low pH state. Okay, so why do I think this is matter? Why do I think this nitric oxide matters? And you can go to my video on ALDH7A1, and this is a slide from that. So I'm going to just give a quick recap. If you're in a high nitric oxide state, nitric oxide, normally this is a normal process. We fluctuate in methylation. So we have a thing called methionine salvage pathway, and usually we use B12 and 5-methylfolate to run this pathway. But anytime that nitric oxide levels go up in the body, that particular enzyme methionine synthase is inhibited. And this enzyme is in our liver, and it's found in the brain. They used to not think it was in the brain, but it is occurring in the brain, and then it also occurs in the kidneys. So there's 3 areas of the body that methionine synthase happens everywhere, right? But there's 3 areas in the body that have a backup pathway called BHMT, or betaine homocysteine methyltransferase. That's another enzyme. So in the brain and the kidneys and the liver, you can use BHMT to form methylation. So there's a backup enzyme. Okay. So if you have to use BHMT to run homocysteine to methionine, then you're going to need a thing called betaine. Okay. And if you aren't eating betaine, then you're going to have to make it using ALDH7A1. And ALDH7A1, also known as antiquintin, is actually a moonlighting enzyme. So if you're busy over here making betaine because your nitric oxide levels are way too high and you can't use folate and B12 properly in your liver, where you metabolize some of that vitamin A, right? And in your brain, which also requires metabolism of vitamin A, and then also in your kidneys, which also is very much involved in vitamin A. If you're not using this enzyme to, if you're using this enzyme to make betaine, then you're not using this enzyme to metabolize lysine. Or you could also be having a lot of reactive oxygen species and tying up this enzyme over here to detoxify a lot of reactive oxygen species. So if you end up pushing this enzyme into all these other 2 pathways, and you're not using it to metabolize lysine, you end up with a lot of P6C, and P6C will bind to B6, P5P, active B6, and make it inactive. So you'll look like you have normal B6 levels in your blood, but it's inactive. I couldn't find, I have not found, we've been looking, my friends and I have been looking for a long time to try to find if they can do a test to see if the B6 in the blood is inactive, or by P6C, but there's not that out there. It's just not available. So I think symptomatically, though, this is making sense. We do know that in liver disease, we see high levels of pipiculic acid, and pipiculic acid and P6C are interchangeable. Basically, it's one or the other, right? It's going back and forth here. And so we have this ongoing issue when nitric oxide levels are too high, that we're going to have bad lysine metabolism if we're not eating betaine. So you can go check out that video. I know I kind of said I was going to go really quick, but I never can go quick on this pathway because I really think it's amazing that this enzyme is moonlighting and causing all these problems. But the main problem here is nitric oxide. Okay, so why do we need active B6 in relation to vitamin A metabolism? It's because we require B6 to be able to finish the kynurenine pathway, and that is where we make our NAD. And NAD is the cofactor for vitamin A metabolism. So you can see if you can't get out of this, what I think is a functional B6 deficiency, you will not be able to make your own NAD in your body to be able to metabolize vitamin A. If you can't metabolize vitamin A from retinol to retinaldehyde to retinoic acid, then you aren't able to detoxify it. You can't get it out of the body. You also can't use it. You can't use physiological active vitamin A, retinoic acid, if you're not making it. So you might look like, on a blood work, you have high vitamin A, but when you present to me, a dietitian, you look like you have vitamin A deficiency. You have keratosis pilaris, you have poor wound healing, you have a bad immune system. All of those things wreak vitamin A deficiency. But if you don't check someone's blood to see where they are with vitamin A metabolism, and you don't check their iron status to see if maybe vitamin A is just like locked in the liver, and you just add more vitamin A to the system, that doesn't help the person. Vitamin A is very much about metabolism, and it requires NAD. So we have to be making that. And to be able to make NAD from tryptophan, you require B6. You also, by the way, require B2. So be aware of that. You can't just be like, it's all B6, because there's two ways to get down here to NAD. You can use B6, you can use B2. So that's important. We have to not have that high nitric oxide level that's inhibiting methionine synthase, and it's causing potentially a functional B6 deficiency if someone's not consuming enough betaine to support that pathway. So, whoops, go too fast. Okay, so what are my solutions? I have a lot of solutions. This video's been in the works for a while. I actually took a break because my daughter got very sick. Also, my brain wasn't working too well. I'll tell you about that in a second. Okay, so the first solution is pH. Working on acidosis, because you don't want to be in acidosis because that encourages spontaneous production of nitric oxide from nitrate. So something you could try doing is holding your breath. I mean, not until you pass out, don't do that, but maybe slow breathing, that helps to restore CO2 levels, or maybe holding your breath. I have had POTS in the past. I still have a little bit of POTS, but it's so much better because POTS is very much directly related to nitric oxide. Okay. And so, before, when I had really bad POTS, one of my solutions to keep from passing out was to hold my breath for a little bit before standing up, and then the CO2 levels in my brain didn't drop, because that's actually what causes people to lose consciousness, is a drop in CO2. The reason why is you have to have physiological levels of CO2 to be able to exchange oxygen across the cell membrane. So holding your breath will help to restore CO2 a bit. You want to avoid a ketogenic diet. Ketogenic diets actually cause acidosis. So it's not going to improve the situation. It's going to cause that spontaneous nitric oxide production. You could try sodium bicarbonate to increase pH, but that could lead to mineral deficiencies because it alters the intestinal pH. So another option is to try citrate forms of supplements, because that can help restore pH. You could try potassium citrate, magnesium citrate, and then one of my friends, Daniel, he said he's been doing betaine citrate. So that's great, because betaine is what we need when we're in this high nitric oxide state. So that's a really good solution. I didn't even know that existed. Learn something new every day. So I think my dog's about to bark. Hold on. Okay, no, we're good. Okay, so the next solution is to reduce nitrates or also monitor. Oh, that was I already talked about acidosis, but reducing nitrates would be really good. So if you're in metabolic acidosis, you want to try to avoid excess nitrates. You would also want to avoid high dose vitamin C. So don't do that. Vitamin C can also inhibit aldehyde dehydrogenase, which is the enzyme that helps you to convert retinoaldehyde to retinoic acid. So you don't want to do that either. You really, really want to avoid very, very high levels of vitamin C. If your CO2 or your bicarbonate is low on a lab test, you could supplement, like I said in the previous slide, with bicarbonate. You could consider decreasing dietary nitrate to avoid excess nitric oxide formation. So you want to avoid things like lunch meats. And again, as I said before, even those labeled nitrate-free contain celery powder, which is a nitrate source. So watch for that. You want to avoid excess celery, beetroots, or the juices, lettuce, spinach, cabbage, arugula, garlic, pomegranate juice, anything that has a lot of nitrates in it. You would want to try to avoid those. So here's a possible solution, benfotiamine. Actually, before I even knew that I was struggling with vitamin A metabolism, I started doing the high dose thiamine protocol, which I am not against, by the way. I'm not against it. It has a place. All of these things that we're doing are band-aids until we can get our normal metabolism back on board. I like band-aids. Band-aids serve a purpose, but we have to find root cause as well. So, like your root cause could be a fungal infection. Your root cause might not be vitamin A, although that could be the thing that broke you. Your root cause could be oxalate toxicity. That was my daughter. Your root cause could be a bacterial infection. Who knows? But in any case, you can use a Band-Aid to help yourself get out of it. So benfotiamine actually counteracts RBP4 and so you have INOS, right? And benfotiamine can actually inhibit the translocation of nuclear factor kappa B to the nucleus. And so that will lower INOS. And it also decreases tumor necrosis factor alpha, which again kind of comes back in and causes even more INOS to increase. So benfotiamine has been shown to downregulate the NFP kappa cascade. So that's a good thing, right? Another thing that it does is it's a carbonic anhydrous inhibitor. So it allows for more CO2 to remain as CO2, which can mop up excess nitric oxide. But that can also backfire. In fact, there's some, if you go on Hormones Matter, you can look for an article about someone who was on high dose thiamine for a while, and it acted like a carbonic anhydrous inhibitor and caused renal, kind of like a renal tubular acidosis, and made them more sick. Some of the medications that are out there for treating migraines, like Topamax. Topamax is a carbonic anhydrous inhibitor, and it can cause CO2 to drop very low as well. So there might be like a Goldilocks zone for benfotiamine, where you have just the right amount in your body to help with your CO2. So it may or may not work for someone. CO2 alone is a really good buffer for nitric oxide. So this could be another reason why many with vitamin A toxicity have low CO2 levels. It might be that they are actually kind of using CO2 to buffer nitric oxide. So yeah. So another really good thing about benfotiamine is it decreases H&E adducts, and so there's less burden on ALDH7A1. So that means that lysine metabolism can go better, and then that means that your B6 will still be functional. So benfotiamine, I think, can help to keep B6 active. And then it actually decreases lipid peroxidation too. And again, that's a place where ALDH7A1 also has to work. So if benfotiamine lowers lipid peroxidation, then you can still make that functional B6. You can have good B6 in your body. One thing to caution with benfotiamine, though, I would say, like, this is my own anecdotal evidence. I've noticed that because it does downregulate INOS, if I get sick with a bacterial infection or a virus, if I'm taking benfotiamine, I don't recover as fast. And I think it's because I'm downregulating the nitric oxide pathways too much. We need some nitric oxide to be able to fight off these bacteria and viruses. Okay, so the next one is betaine. So betaine is great because if you consume betaine, then you don't have to worry about using that ALDH7A1 enzyme, right? And that means that your B6, in theory, won't become non-functional from P6C. But betaine also inhibits, or possibly inhibits, NFKB translocation to the nucleus. So this would also decrease INOS. And it helps to restore, like I was saying, I think it helps to restore B6 by helping ALDH7A1 to be able to work on lysine metabolism instead of being over here making betaine. If you have normal B6, then that will lower your histamine levels because you need B6 to get rid of histamine. And that means less triggers for nitric oxide because having high histamine levels increases nitric oxide levels as well. And then if you have, if you're taking betaine, then you'll have a normal B6 status. And then the kynurenine pathway can continue forward and you can make NAD and that can help you metabolize vitamin A. So betaine is very helpful. I myself, my family, we use a betaine supplement. We use trimethylglycine. We use smaller amounts than what's on the label. The label is like one serving is 1,500 milligrams. And we use like, for the littlest one, she gets 250. I take 500. So again, talk to your own healthcare provider about how much you need. A side effect of betaine is if you are really stuck in BHMT, you can end up with high sarcosine levels. And, you know, there's a lot of good studies about sarcosine, how it's good for your mood. I mean, I'm in a good mood, but I have high sarcosine levels, and sarcosine, in theory, in my brain, I've kind of looked around for all this stuff. Sarcosine can chelate copper. And so a copper deficiency is going to trigger some really bad stuff in the brain. You won't be able to use superoxide dismutase. And so you'll end up with a lot of superoxide. And so for the past few months, I went downhill like really bad. And it was because my copper tanked. So I think that I didn't have the nutrients on board to deal with sarcosine, and it was basically my riboflavin was probably too low. That was my conclusion. So with betaine, you have to be careful. I have a different video about this. It's in the ALDH7A1 video. You should definitely get a plasma amino acid level checked before you start trimethylglycine. Because if you are the rare person who has a slow CBS gene and your methionine level is already high, you could cause brain edema. And that's not good. That means bad neurological problems. So I always have everyone that I work with, I encourage them to get a plasma amino acid test before they start this. And also you could just do very low dose, or you could work on dietary betaine. That's a possibility too. I'm just not able to do that because our family is gluten-free. And also we're low oxalate, so that kind of keeps us from eating betaine. All right, next thing, a solution would be to monitor your heme production. So some people are really bad at making heme. These are people with porphyria. So there's tests out there that you can do. There's a urinary porphyrin test to see if you have porphyria. It's kind of common in this world of unhealthy people. So it might be that you're not an outlier, you're just one of us. And if you're not making very good heme, then you're going to be bad at buffering nitric oxide. Also, iron is where the nitric oxide binds to heme. So it can bind, it's one of the places on heme that it can bind to. So if your iron levels are low, then you're going to have problems with nitric oxide. So you might try checking ferritin, TIBC, transparent serum iron, and complete blood count. Whenever you check iron, you want to check all these parameters just to see where you are, and then, have a health professional look over it with you and talk to you about what's going on. One thing that's important to know is I am not a fan of high dose vitamin D, not at all. If you're on high dose vitamin D, it'll plummet your ferritin. You won't be able to make ferritin. And so what you'll end up with is a large amount of serum iron, which is not good. So that's something to look for. Ferritin is also low during an inflammatory state. So it might not be a really good indicator of your iron stores if you have elevated liver enzymes. So you kind of have to go based on symptoms too. So like if someone were to have restless leg symptoms, but also elevated liver enzymes, I would think that they actually do have an iron deficiency, at least a functional one. So there's that. Also, if you are bad at vitamin A metabolism and you have low retinoic acid levels, that actually increases or keeps you from being able to downregulate the hormone hepcidin, which blocks iron absorption into the body. Hepcidin is also made by fat cells. So if you happen to be overweight, it could be that you have lots of hepcidin blocking your iron absorption. Vitamin D can downregulate hepcidin. But again, I'm not a fan of high dose vitamin D. So you would need to get like a squirty lamp, or if you live in the country like me, you can sit by a nice window. It's kind of dark today. It's not very sunny. So that's important though. It's important to have normal iron status to be able to buffer nitric oxide in your blood. So all of our body systems have to come on board to be able to deal with these things, right? So another solution is copper and zinc. They're both buffers of nitric oxide. So when you're checking labs for copper and zinc, you want to check serum copper, ceruloplasmin, which is the carrier for copper, plasma zinc, alkaline phosphatase, because alkaline phosphatase kind of tells you like if you're okay on zinc. If your alk phos is low in the blood, then likely your zinc is still not optimal. You really you have to work on that a bit. And the reason why I say that is that it's hard to look at blood levels of these particular nutrients, because if you're in an infection state, then your body actually sends copper and zinc out into the blood. So you can't just look at serum copper, you have to look at ceruloplasmin also. But in any case, if you're in some type of infection, these levels might look higher in the blood than they normally do. So blood isn't the best way to look at it. But then hair testing, gosh, that stuff confuses me. I'm not a hair testing expert at all. So what do I do? Like with my own clients is I look at dietary intakes. I look at, have you been consuming zinc? Have you been taking a zinc supplement? Have you been eating copper foods? Has your diet been super limited over the past 2 years? And then I could try to extrapolate whether they actually have an issue or not. If they have low neutrophil levels, likely they're copper deficient, because you have to have copper to be able to make these neutrophils. So, and then if their alkaline phosphatase is low, I would think that they have a zinc issue. I probably should have put like a complete blood count, CBC, on there as something to check too. So zinc itself can inhibit INOS expression. So it can just kind of, it has a feedback mechanism. I'll show you a slide in a second. If you're going to supplement with copper and or zinc, you want to be aware that taking more than 40 milligrams of zinc for longer than 2 weeks can actually induce a copper deficiency, at least a functional one. Not sure about the stores of copper that a person has. There's a disease called Wilson's disease, so it's important to talk with your healthcare provider about your copper status before working on copper. And also, here's a problem. Even though zinc and copper can both buffer nitric oxide, copper induces INOS. So going really high on copper increases nitric oxide levels. So try not to do high dose copper. If you have done it, consider cutting back on it, because it is a known inducer of INOS. Again, I think it's more of a Goldilocks nutrient. And then if you're going to supplement with copper and zinc, you could work on doing like a 10-to-1 or 15-to-1 ratio. Everyone varies on what they think is the best for that. And be aware that bicarbonate treatment can alter your mineral uptake in general. So usually it's iron that's effective, but I myself found that copper was affected, just experience. So, okay, so here is a slide showing what zinc does. So whenever nitric oxide, so you have your iNOS and mRNA, and you have iNOS here, and you're making nitric oxide. So the nitric oxide actually induces zinc release from zinc clusters of intracellular proteins. So the zinc actually is like discharged from the proteins that are within the cell, and then the zinc goes in and has a feedback regulation on nuclear factor kappa B. So it's sort of like amphetamine. So zinc can be helpful, and I've heard from my friends that people who have hypervitaminosis A in this other group, they use zinc and they find that it's very helpful. Again, just monitoring for copper deficiency symptoms would be an excellent thing to do too. And now B12, this was really interesting. So we've all heard that B12 mops up nitric oxide. Like that's like been something that people have talked about for a while. So I went on a literature hunt and I'm like, okay, so hydroxycobalamin could maybe be a buffer for nitric oxide. That's cool. But then I found that hydroxycobalamin actually inhibits iNOS. So that's so cool. So here, whoops. So here is a table from a journal article about this. And here's hydroxycobalamin, and this is nitric oxide synthase 1. And here's NOS3, and iNOS is NOS2. So NOS1 is kind of ubiquitous. It's all over the body. The heart uses NOS1 to be able to make nitric oxide in the heart muscle. And then NOS3 is endothelial nitric oxide. So here's iNOS, the inducible form. And this estimated ED50 is the point at which, in micromoles, the form of cobalamin inhibited iNOS by 50%. Okay, so at 21.1 micromoles, hydroxycobalamin inhibited iNOS by 50%. But also know that it inhibited like the normal nitric oxide synthesis that's kind of working normally throughout the body at even a smaller amount. That's important to realize. But if you look down here, methylcobalamin, it took, if in no light, so this is methylcobalamin exposed to no light, it took 231 micromoles to inhibit iNOS by 50%. But if you expose the methylcobalamin to light for 60 minutes, it only takes 63 micromoles to inhibit iNOS. And then the same thing kind of happens with adenosylcobalamin, but it's like crazier because adenosylcobalamin that's been kept in dark, when you add that to this assay that has iNOS in it, it takes 2,148 micromoles to reduce the enzyme activity by 50%. But if you expose adenosylcobalamin to light, then it only takes 60 micromoles to inhibit iNOS by 50%. So the researchers said that if you expose the adenosylcobalamin or methylcobalamin to light, it produces hydroxycobalamin. I was so shocked by that because literally my bottle of methylated B12 or methylcobalamin, it says to store in a cool, dark place. And okay, y'all, comment in this video if you know if something toxic happens when it's exposed to light, because I don't know, like do your own research. But what if I can save so much money by just pouring my methylcobalamin into a clear jar and letting it be exposed to light before I take it, and it'll change the potency of it. I don't know, possible. So that was shocking to me. But again, do your own research because I haven't researched this fully. I just came across it today, and I don't want anybody getting hurt. Um, so that's really interesting. You can use hydroxycobalamin to inhibit INOS. And then there was this other study that I will link in the description where they can use hydroxycobalamin in a state of like septic shock, kind of in a really bad, like an acute care setting. And it actually regulates INOS. It doesn't completely knock it out. It just makes it more manageable. And so hydroxycobalamin seems to be like the best version of B12 to use when your high nitric oxide levels are an issue, because it is more of a regulator of INOS and doesn't necessarily knock it completely out. Whereas benfotiamine is going to knock that sucker down. And so like benfotiamine is not the one to be using when you're sick. But if you're sick, you could still continue your hydroxycobalamin because it just helps regulate not too much INOS. There's enough going on to have a normal inflammatory response, it seems like. So that's really good. Okay, so I think that's the end of my presentation. Those are all the solutions that I have for right now. Just remember that as vitamin A levels go up in that blood, you're inducing INOS, and INOS is increasing nitric oxide levels, and nitric oxide, not good. We don't want that. And I haven't even touched on the fact that if you're pushing INOS constantly, you can also be using up this cofactor called BH4. And once you're not using BH4 correctly, like not coupling INOS with BH4, then you end up with even worse byproducts like peroxynitrate and superoxide. So it's super important for us to not have too much INOS going on at any given time in our bodies. There's something else I was going to say about that. Oh, goes along with people who have seizure disorder, which is really common among people with hypervitaminosis, because you have to be making retinoic acid in the brain in physiological levels to control nerves. It's physiological levels, not toxic levels. So there's another medication out there. There's DHFR inhibitors. So like lamotrigine, I'm trying to think of another one. Crap. I don't know. If you look up DHFR inhibitors and seizure medications, lamotrigine is definitely one of them. And it can inhibit DHFR, which is needed. It's one of the pathways needed to recycle BH4. And so there's a risk there that if your nitric oxide levels are high and your BH4 is low and you have uncoupled nitric oxide synthase, then there's even more damage happening. So it's really important to get this nitric oxide stuff under control. And that is the end. And please let me know if you have any questions or comments. I encourage that. Just one brain here. I'm not a super genius. I'm just a person who has way too many experiences and has family members suffering, and I myself have suffered. And so experience makes experts. And that's what I know about everyone watching this video. You all have your own experience. You're all experts in one area. And if we can come together, we can work this problem. We can all get better together. All right. Have a great day. Bye.